nicer on the stain. It's a little darker. And right here will be portal triad. So when you're looking at a distance, a lower magnification on a portal triad, you're going to see sort of this clearer area around it, where it's like a little bit lighter in color. So that's kind of a good standout there. But when you blow it up, you're going to start to see the components of the triad. And as the name suggests, it's a triad, so there are 3 parts to this whole thing. Okay? And here they are. So maybe you have a portal triad. You have a venule, which you can see right here. It looks like the veins that we looked at before, right? It's got a thin, smooth muscle wall, and it's kind of irregularly shaped, usually a little bit flattened. Okay? And then you can see this guy right here has a thicker muscle wall around it, smaller than the venule, it's rounder. This right here would be an arteriole. And this one right here, you can see the little dots of the nuclei lining the outside, the inside of it right here. It's made a simple cuboidal epithelium. This would be a bile duct. Usually bile ducts are a little smaller than arterioles, not always, but you can always, they'll always stand out because of those nuclei, making little dotted edges around the inside. Okay. So portal triad, 3 things. Venule can be larger, usually the largest of the 3, sort of irregularly shaped, thin, smooth muscle wall. Arteriole is usually going to be very round, thicker, smooth muscle wall. Sometimes you'll see red blood cells on the inside. You can see red blood cells on the inside of the venule here. And then the bile duct is going to be lined with simple cuboidal epithelium, and you'll see the round nuclei sort of around the inside. Okay. Now, they also list Kupfner cells and hepatocytes. Hepatocytes are what make up pretty much everything else. You can see all the darker pink stuff, and I'll zoom out and makes up most of the liver. Those are the hepatocytes. The Kupfner cells, they're kind of like the oxyphil cells, that we looked at before. They're kind of hard to differentiate from the Kupfner cells, so I won't really expect you to be able to do that. They don't even really know the function of the Kupfner cells. Okay. So let me pull it out one more time. So if I point to just all the, just the pink stuff, hepatocytes would be what I would expect. I'm not going to expect you to differentiate Kupfner cells from them. Okay? Now, last thing is what they call central vein. That's a good one. All right, I guess. All right. I wonder if I really do. The one I actually like the most, you can only see part of it here. Sadly, was kind of cut. But you can see this huge vessel running through right here. It's got a fairly, compared to like its overall size, it's got a pretty thin muscle wall. And you can see right here, it's filled with red blood cells. That would be a central vein. It doesn't have that really nice light colored layer around it. It's got a little bit of a clear layer, but not as defined, not as light colored as around the triad. Let me see if I can find, there's a little bit of a smaller one that's all right. It's over this way. I like that one personally. There's another portal triad, another portal triad. Here would be another one of those central veins. This one's not as pretty, but it's all right. You can see it's huge, right? Fairly thin, smooth muscle wall, and you cannot, as you can see, there's not a bile duct or an arteriole associated with it. It stands alone. Okay, so central vein is going to be large, thin, smooth muscle wall, irregularly shaped, but what sets it apart from a triad venule is you don't see, it's not associated with a bile duct or an arteriole. All right? So those are the parts we have on the liver. Next one we have is the pancreas, which I'm sure you guys remember from before, right? I told you it would come back to haunt you. That wasn't it. Someone said it was a good slide, but Here, let me grab the slide from out on the scope. Or I can try this one. How about this one? Give this one a shot. This one's going to be purple in color instead of pink, but it's still going to look the same in terms of texture. I also have all the slides out on the scopes. So what I'm going to be eventually doing for your participation grade is going to be related to the scopes. So you will have to look at it. Here, that's pretty bad. See if I can bring this a little bit. Yeah. There we go. All right, so remember before when we talked about the pancreas, what did we really focus on? The endocrine portion, which is in the what? The islets, right? So if there would be an islet, the islets are going to be lighter in color. In the case of the one that's out on the scope, that makes up the acinar cells, which are the exocrine portion, are going to be dark pink. The islets will be a lighter pink. Here, the islets are light purple. The acinar cells or acinar cells are dark purple. But other than that, it's the same, besides the color. Okay. So the islets of Langerhans are the ones that are exocrine. What do they produce? I'm sorry, they're endocrine. Yeah, what do they produce? Insulin, right? And what about everything else? The acinar cells. Lipase, right? Which is a digestive enzyme. Okay, so that's exocrine. Okay, so islets of Langerhans, everything else, the darker stuff, acinar cells. Okay. Or acinar cells. Whatever, what's your vocabulary, what it has. Okay. Last one we have is the large intestine. And it's actually quite a bit simpler than the small intestine. So let's go ahead and give that a look. So with the large intestine, you don't have fleet line on the inside like you do the small intestine, but you can still see you have these big folds in the walls. But they don't really name the folds here. So this is the large intestine. And you don't really have much digestion happening in the large intestine, but you have the production of vitamin K and also the absorption of water, right? You can see between the folds in the small intestine, or in the large intestine here, you have these big kind of gaps between those folds. Those are called crypts. So like, you know, old show Tales from the Crypt, spelled the same way. So these big folds, the spaces between the folds are called crypts. So you don't have any villi here, you don't have any plecos. You just have these crypts. Okay. Now, let me blow it up. Lining the inside of the large intestine, we have, once again, simple columnar epithelium, but embedded with these cells, you can see the nuclei are kind of staying this dark purple dots, but you can see these sort of more pinkish colored cells that are kind of shaped like a glass of red wine, kind of the same color. These right here are goblet cells, and they produce the mucus that lines the inside of the large intestine. So since the large intestine is absorbing a lot of water, that means the contents are going to be more solid. So in order to facilitate them moving smoothly through the large intestine, that mucus is really helpful.